Okay, I'm going to do a pretty quick one today, so hopefully I can get this cranked out very quickly. So last time I had gone over some of the alignment of multiple SARS coronaviruses with multiple POMCs from different mammals, and that was a way to look at a lot of conserved amino acid residues that are going on between the different coronaviruses and that are also conserved in the POMCs. But what I want to do right now is just focus on the SARS coronavirus or SARS-CoV-2 because SARS-CoV-2 is behaving differently from other coronaviruses. So if there's some new pathology that is the result of it, we want to look at what are the different amino acids, or are there regions of different amino acids. Now, granted, uh, what a lot of viruses can do is they can have a lot of amino acids set up, but only change one or two, and then it completely changes the the entire amino acid sequence's role or its abilities. So you can have knockout mutations or gain of function mutations. So a virus can have pretty much 99.9% of the machinery to do a particular task, but if a key amino acid residue gets mutated, then the whole thing could be useless, but that might be beneficial for a virus to survive longer in its host, for instance. So you can't just look at the amino acid sequences and say, yes, it is definitely doing this or it's definitely not doing this. However, we're going to be focusing um, a lot of our work on, or our focus on peptides, where especially the way that your antibodies are produced against certain peptides. If there's a lot of homology between them, then there's going to be a lot of crossover between them. So you can be a little bit more liberal with how an antibody is going to cross react and affect versus the straight up molecular, what would the word be, function of a particular amino acid sequence. So here, as you can see, I did an alignment with the other spikes and the other uh, peptide hormones. And over here, in the beginning of a lot of the spikes, there really isn't that much homology between spike of SARS-CoV-2 and the other spikes. There is some, but it's not that great. Over here, you start seeing some, but they really have a hard time lining up the residues over here. So what I did was I did the same thing with one with the spike of SARS-CoV-2 and the other proprotein peptides. And here we see an alignment and a pretty damn good one. So what I wanted to do was look, does this peptide or does this peptide, which is very similar to it, does this show up in the um, does this show up in the literature at all, or do similar sequences show up in the literature? And lo and behold, oh, and I should also mention that this sequence, some of it is exposed to the outside, so it would possibly be antigenically important. You might possibly be able to make antibodies against the sequence because this, where is it? Okay, this asparagine is glycosylated, so that would indicate that it is on the surface of the spike protein. So right in the middle here, we know that this amino acid is exposed, which would indicate to me that a lot of it is also exposed. And I do believe as well, this cysteine might have a disulfide linkage. So so that system might be pointing inwards or doing something. I'm not going to focus too much on it, but that disulfide linkage might change things a lot. So it might make this very dissimilar from the native peptide in your body, but uh, it is something to consider. It's still going to be a very similar peptide, but this cysteine could change things significantly. And I don't know about this cysteine in the final peptides, but I do know that this cysteine in the proprotein is there's a disulfide linkage there too. But I presume that it gets broken up when the rest of the peptide gets broken up as well. But don't quote me on that. Regardless, I decided to look up the sequence and let's see. I need to get to, where did it go? Aha, here. So as I said earlier, I had to go to the peptide database, and that's a completely different one. I had to go to the peptide database, and here, this is the isolation and characterization of two peptides with prolactin release inhibiting activity from porcine hypothalami. So there was a period of time where a lot of research was going into what were the hormones that were mediating the inhibition of the release of prolactin, which is a peptide hormone that is pretty darn important. Um, Wait, let me double check to see if it's a peptide hormone. Images. Oh, I don't want to go here. I want to go to Wikipedia. Luteotropic hormone. Yeah, so it is a protein or a peptide. Yeah, so it's a protein or a peptide hormone. Regardless, it is released, and we spent some time trying to research the type of or what compound in the body prevents the release of prolactin into the body. What stops it or what inhibits it? And we now believe that dopamine is the main contributor to preventing the release of prolactin. So if you have more dopamine in your body on average, or if you get a spike of dopamine in your body, it's going to cause a inhibition of the release of prolactin. So here, isolation and characterization of two peptides with prolactin release inhibiting activity from porcine hypothalami. So these people took the hypothalamus of a pig or pigs, and what they did was they basically blend it up and then they run it through filters, and the filters are going to separate the different components the of the of the mushed up brain bits or the hypothalamus bits. And as things get separated based on their size or their chemical properties, they end up getting put into different aliquots or different pools. And what they want to do with those pools is see what type of activity metabolically these pools have. So it's a way to, if you want to test, it's really hard to remove confounding factors in biology and biochemistry. So one way to remove the confounding factors is to just separate everything out as much as you can. And then if you find activity in the bits that you in certain areas, then you can say, okay, look at this particular pool or this aliquot or this particular portion of the organ that I just blended, and this has the activity that I want to find. So then you go into that pool and then you look at what's in that pool. So here, I'm just going to read this. Two peptides with in vitro prolactin, in vitro meaning in a test tube or in glass, prolactin release inhibiting activity were purified from stock median eminence or SME fragments of 20,000 pig hypothalami. Monolayer cultures of rat anterior pituitary cells were incubated with aliquots of chromatographic fractions and the inhibition of release prolactin vitro was measured by RIA in order to monitor the purification. So, monolayer cultures of rat anterior pituitary cells. Basically, this is a cell line from rats. So it's a model organism. It's not exactly humans, but there's a lot of crossover between what works in a rat brain and what works in your brain. And biochemically in general, a lot of stuff that happens in rats is going to happen in you as well. So here they have a cell line of the pituitary cells of rats, and remember the pituitary is a gland that regulates a lot of hormones in your body. Um, so they were incubated with aliquots of chromatographic fractions. So chromatography is basically when we separate anything. It comes originally from the Greek word chroma or chrome, basically, which means color, because if you separate, I don't know why, what the exact experiment was or the exact etymology was, but basically you can separate colors. And if you do separate colors, they will form rainbows or a bunch of different colors. So if you have like a marker, for instance, and you put that make a dot with that marker, and you let water run through it, the individual inks of that marker bit are going to separate from each other because they all have different characteristics. So that's where the word comes from. So aliquots are different pools of the chromatographic fractions, and these cells were incubated with those chromatographic fractions. So whatever was left in that pool when they separated a bunch of stuff out gets mixed in with these rat and pituitary cells. And then they measured the release of prolactin in order to monitor the purification. The hypothalamic tissue extract was separated into 11 fractions by high-performance aqueous size exclusion chromatography, with one fraction showing a fourfold increase in prolactin release inhibiting factor activity. So this material was further purified by semi-preparative reverse phase RP high-performance liquid chromatography. So basically, they found a aliquot that had fourfold increase in prolactin release inhibiting factor, and they don't know what that factor is. They're just looking for any chemical that has this sort of characteristic, releasing or inhibiting the release of prolactin. And then they further purified that one fraction through a more high-tech, high-pressure form of chromatography, usually with a smaller tube. And this process resulted in the separation of two distinct fractions that showed high PIF activity. These were further purified by semi-preparative and analytical RP-HPLC to apparent homogeneity as judged by UV absorbance profile. So they took it and they just kept on purifying it until they could get the exact peptide that they were looking for. Because a problem with doing these purification things is you can purify some stuff, but it's still noisy. So when you have something that's very pure and very chemical identical, you would normally have you would see it as a very stiff and a very unnoisy normal distribution. But if it's impure, oftentimes you'll see a wider distribution and a lot of noise within it. So you just got to keep on purifying it and finding different ways of tweaking your machine so that you find the right way to separate the protein or the peptide or whatever chemical you're looking for from the other chemicals in that solution. So they just kept on doing it until they were able to get the clean looking peaks. Continuing, and that's what they mean by apparent homogeneity, is it looks homogeneous. So neither of the two peptides showed cross-reactivity with gonadotropin releasing hormone-associated peptide or with somatostatin 14 antibodies. Protein sequence analysis revealed that one of the PIFs, and I forgot what the prolactin release inhibiting factor, so one of the PIF peptides was tryptine, cytosine, leucine, glucine. So I'll just say it: RCLGSS. Glutamine. That is a Q. Yeah, Q C. Well, we'll just go back to it. So I believe this is the sequence. Let me just double check because it gets hard to remember all the amino acid sequences sometimes. So let's just look up SS. Oh yeah, so it was a Q. So yeah, that's why I was having a hard time. So as you can see here, this would be a W, C, L, G, and you can see here W, C, L. Oh, did I say W, C, L, G, S, S? Oh my gosh. Yeah, glutamine is E. I forgot about that. I was thinking glycine for a second. So this is the peptide, and identical to residues 27 to 52 of the N-terminal region of the pro-opiomelanocortin precursor. So remember, this protein, this chole_human, this is the POMC, or pro-opiomelanocortin precursor, and this is the MPP peptide. It's called the NPP because it's the N-terminal, what's it called? N-terminal region of the pro-opiomelanocortin precursor. So NPP, and this is a portion of it. So this isn't the complete peptide. So this is somehow a piece of that large peptide. So it could have been just from normal metabolism, the peptides are breaking down, or it could be a very important, it could be playing a very important role in homeostasis of the organism, in this case, the pig. But we would assume that it would also be working in you as well, or at least a similar peptide would be working in you. So identical to residues 27 to 52 of the N-terminal region of the pro-opiomelanocortin, or POMC precursor, and the sequence of the other PAF was this, and I believe, yeah, and representing residues 109 to 147 of the vasopressin neurophysin precursor, which is a completely different protein peptide. Synthetic peptides corresponding to the N-terminal region of POMC had significant PIF activity in vitro. So they didn't just check out these peptides. These peptides told them, okay, this is what's working. But what they're saying is they also made other peptides corresponding to the N-terminal region of POMC, and they had significant PIF, or the prolactin inhibition factor activity in vitro. So now let's bring this back to this particular sequence. So we know that one of the strategies that viruses will employ to evade the immune system is to have epitopes, or pieces of the virus, look similar to proteins that are within your own body. Okay, so and one of the reasons why this is important for a virus is if you make an antibody for that piece, that antibody will not be very effective because, for one, it's going to be attacking your own body. So a lot of those antibodies are going to be used not to attack the virus, but they're going to be sequestered by attacking your own body. And it's very effective for a virus to make antibodies against your peptide hormones, because you have a lot of those floating around. right? So here, this is what worries me and jumps out to me when I look at this. I know that this is an exposed region and possibly antigenically important for your body. This is exposed on the spike protein, and so you might be making antibodies against this particular region. Now, what I want to say is, of this particular region, you have one, two, three, 4, 5, 6, 7, 8, 9 absolutely identical residues within a 14 amino acid region, and then a lot of the others are very similar. So Q versus NN, so glutamine versus asparagine, which amide residue, pretty much exactly the same. Threonine instead of serine, it's another alcohol amino acid. Yeah, so as I look at this, this tells me that if you are making antibodies for one of these, you're probably going to be making antibodies for another. Now, granted, I should mention that your body, this asparagine right there, your body will glycosylate this asparagine, so we know that much, and that asparagine can make this look different enough to maybe stop some of the cross-reactivity of antibodies. But granted, that also means that that doesn't always happen. Glycosylation isn't always going to happen, so that means you'll probably have plenty of unglycosylated versions of this peptide floating around. So that is something to be considerate of, and especially if your body starts, degrading these spike proteins. A lot of them, you could have a lot of peptides just floating around, and a lot of the sugars could just hydrolyze off as well. So this would end up looking very similar to this. Or even if it doesn't look very similar, or even if it does make an antibody that glycosylates this area, this is a free-floating peptide, and I don't believe it has a lot of cysteine or disulfide linkages in it, which would help it maintain its shape. But even if you make an antibody for this, and that antibody secondary structure is, or primary structure, is different from this particular peptide, what could happen is maybe a piece of the peptide will bind to the antibody, and then the rest of the peptide will then curl into the secondary structure of the spike protein. So even if it's not secondary structure-wise an exact match, the effect of the antibody could cause the peptide to go into that other configuration. So that's just something that we need to be mindful of, is your body is probably, if you get SARS-CoV-2, it's probably going to affect the concentrations of this peptide in your body. I'll be right back. Do you want out? So what does this mean, physiology, or what does this mean for your physiology? Well, we know that this peptide, if it's floating around in your body, and I shouldn't say because it was implicated in mice, but we can pretty much extend that to humans. If you make antibodies for this, your body's going to start removing the antibody out of circulation, or start removing the peptide out of circulation. And that means, since it is a prolactin release inhibiting factor, then your body's going to produce more prolactin. Okay? And if your body produces more prolactin, that can have serious implications on your immunity. That could have serious implications on your, I'll have to look at this. I believe prolactin causes the upregulation of ACE2 receptors. So I should look into that to double check. But if that is true, if my memory is serving me correctly, if your body starts producing more ACE2 receptors, then that's going to be very beneficial for viruses that are looking for ACE2 receptors to get into cells, right? So that would be one thing. Another thing would be just general lowering of sex hormones, because a lot of sex hormones um, are suppressed by high levels of prolactin in the body. So that could just be that's bad for men and women. Like that's not it could disproportionately affect men, but and that is what we see. But yeah, just keep that in mind. If this is actually working to sequester these prolactin release inhibiting peptides, then in your own body, then you're going to start producing more prolactin, at least for a period of time. You'll produce a lot more prolactin, and then that could have some serious implications on what sort of receptors your cells produce, what type of immune responses your body wants to have, because your sex hormones play a very large role in what um, basically the inflammation and your immune response. So yeah, just keep that in mind. I would recommend, if it's a cheap test, that you if you do end up contracting SARS-CoV-2, that I would recommend having your hormone levels checked and having your prolactin levels checked, because the virus might be using it to play a role in its pathology and use it to take advantage of your weakened state. So that's all I really want to say about this. I had other papers that I wanted to go over, but it's already late. So thanks for listening. Um, hopefully, hopefully I can get more of this stuff out. I think my next video, I'm going to go over what prolactin is, what prolactin does in the body, and what would what would happen if this is the case. How would it help the virus survive a little bit better in your body? And also we might discuss some of the long-term health issues that would be associated with having elevated prolactin levels for an extremely long period of time, because your immunity is your immunity, and if your immune cells are constantly being um, activated to attack yourself, it's not like so for instance, if you have an allergy, or if you are, I'll just say an allergy, if you're allergic to something, your immune response will diminish once you remove the antigen from your diet or from your lifestyle. So let's say you're allergic to pollen. Well, if you remove all pollen from your environment, eventually your immune system is going to ramp down because it's not receiving the stimulus to ramp up. But if it's a compound that your own body makes at all times of the day, it's very difficult for your body to ramp down its antibody response, or it's harder, um, which is why autoimmune diseases is such a really large growing field. So with that, that's all. Leave any questions, or if you have any recommendations on what topics I should cover, please leave them down. Yeah, okay.